Good evening, everybody. We've still got a few people settling in. It's not the easiest of evenings to get here tonight with the way the weather's been recently. So my name is John Brubaker. I'm a biology faculty member here at CMU. And on behalf of the Scientist in Residence Committee, I'd like to welcome all of you here and thank you all for coming out on, well, it's not the coldest day that we've had or the snowiest, but it is cold and snowy. So it's good to see all of you out here tonight, here at CMU in Winnipeg on Treaty 1 land. This is our 5th annual Scientist in Residence lecture. It's an annual event where we invite a scientist from well, various places. I'll introduce this year's iteration in just a second. To come and speak to us in the CMU community on issues that connect science with Christian theology in some way. And we also, we lean on our scientists in residence to come out and not only speak to us, but also to eat with us, have coffee with us, especially to talk to students and interact with students. And it has been and will continue to be a wonderful program. On that note, the committee and myself would also really like to thank Richard Penner, whose financial support makes this program possible, along with a number of other things that we do here at CMU. So thank you, Richard, and it's always good to have you here on campus. I guess without further ado, I'll introduce our scientist in residence this year. This year's scientist in residence is Dr. Dennis Venema. Dr. Venema comes to us from Trinity Western University in Langley, BC. Given the weather that we've had in the last little while, we're not ungrateful that when he landed on Sunday night, he didn't just turn around and get right back on the next plane to go back to Abbotsford. He also had an invitation to a conference at in Florida this week. So that gives you a sense of the kind of person that we're dealing with here and the sacrifices that he has made. Dennis is married to Valerie and has 3 children aged 9 to 15. He plays guitar, and we've heard him do that here this week. He loves a variety of outdoor pursuits and also a good soak in a hot tub at the end of the day. Dr. Venema holds a PhD from the University of British Columbia, where he studied under the supervision of Vanessa Auld. There he worked with fruit flies as a model system to understand the basis for pattern formation and development of complex organisms. How does an animal emerge from the division, migration, behavior, and contortions of a single fertilized egg and its cellular progeny? Moving from UBC, he joined the faculty at Trinity Western, where he has taught for the past 15 years, and continues to explore these sorts of questions and others with his students. From 2011 to 2018, Dennis was a fellow of biology at the BioLogos Foundation, the mission of which is to help the church and the world to see the potential for harmony between evolutionary science and biblical faith. It's clear that that experience has shaped him deeply, and also that he has contributed substantially to that foundation's important work. If you read Dennis's recent book, Adam and the Genome, which he co-authored with Scott McKnight, and also when you listen to him present tonight, one thing that I predict will stand out is his effectiveness as a communicator and a teacher, a person who seamlessly interweaves stories, analysis, and interpretation. Indeed, Dennis won the 2008 College Biology Teaching Award from the National Association of Biology Teachers in the United States, one of 2 Canadians to have won that award, which is kind of a big deal. He's committed to rigorously following evidence where it leads, but he takes others along with him in a pastoral spirit, with humility and grace. Dennis, I can't even see where you're sitting. That explains why I'm not looking at you at all as I'm saying, okay. It's been really wonderful to have you here this week. We want to thank you for being here this week. I thought you were somewhere back over there originally, but anyway, I should have followed the blinking light. We really look forward to hearing more from you tonight, so thanks for being here.
Thank you for that very warm welcome. It has been such a wonderful few days here at CMU. I've so enjoyed being part of this community for this time, meeting faculty, meeting students. It's just been absolutely wonderful. So thank you for that very warm welcome in contrast to the not so warm weather outside. Would you join me just in a brief word of prayer before we get started? Father God, it is so good to be together. Thank you for your Son, Jesus Christ. Thank you for your witness in creation. Thank you for the gift of intellect and reason to investigate what you've made. We ask that you join us tonight and that you be present with us as we explore these topics, and we give you all thanks and all praise. Amen. All right. You mentioned a good soak in a hot tub. I've found that that's a very useful remedy to getting the Winnipeg cold out of my bones after a day here. You know, us BC types, we're not used to this sort of thing, right? So Florida would have been lovely, but actually I think I've had more fun. I know I've had more fun here than I would have had in Florida, so it's great to be here. So tonight what I want to do is spend some time talking to you about these topics: Adam, Eve, evolution. If you're like me and you grew up in an evangelical setting, those words may have, and maybe for those of us who are a little older than some of the younger students here, those words may, have carried some baggage. I can remember as a young person hearing the word evolution whispered maybe at church, and that was a bad word, right? That was like hearing somebody swear, right? Darwin, oh, it's a bad word, right? And that was the culture that I grew up in, where I sort of just learned by osmosis that if you're a Christian, that means you reject evolution, because evolution is what atheist scientists think up as a way to explain away the obvious grandeur of God's creation. So that is my background, and I'm sensitive to that, and obviously I'm not there now, but that's where I came from. So, and maybe that's part of your story too. Or maybe you're still sort of wondering, what about this evolution business? Is the evidence for evolution any good? Is it a reasonable inference based on the data that we have? So that's sort of the questions that I deal with fairly frequently at Trinity as a person who teaches biology, as a professor. It's been a delight at Trinity Western to be able to teach students material about evolution. And of course, that often generates sort of theological questions. Now, I'm going to have to apologize right from the get-go. Most of what you're going to get tonight is science. I'm going to have to punt a little bit to the theologians, but you'll notice that I didn't write the book by myself. And the reason for that, of course, is that I didn't want the theological side of the book just to be the amateur hour biologist trying to do theology. We all know what we mean, right? Amateur hour theology. So Scott McKnight is a New Testament theologian. I wanted a New Testament scholar on board for the book because often these questions about Adam, Eve, and such often turn to the question of Paul in the New Testament, his use of Adam, those sorts of things. So that's just all to sort of prime the pump there that you're going to get the science half of the talk, but I'll point you to resources at the end where you can sort of complete the picture by hearing Scott in a recorded lecture if you so choose. So let's start by talking about this word evolution and what we mean by that. One of the things that's often a surprise to my students as they learn about biology and evolution for the first time is that they learn that evolution is a theory in the scientific sense, rather than in the popular sense. If you're on Facebook and you see somebody saying, okay, so it shows my age, right? I'm on Facebook, but anyway. Somebody says, well, that's your theory. They're meaning that's your guess, that's your conjecture, that's your hypothesis. They're not meaning that's your broad explanatory framework that has been worked on for 100 years that makes accurate predictions about the natural world, right? That's not what they're saying. So there's this difference between the sort of colloquial use of theory and what scientists mean as a theory. And a theory, in a scientific sense, is actually a very powerful entity. It allows us, it's an explanatory framework for why the facts are the way they are. Science is all about going out and collecting facts about the natural world and then attempting to explain why those facts are the way they are. And a good theory is one of those things. Now, scientists never feel that a theory is proven. Scientists always recognize that we have simply yet to falsify a good explanatory framework. Scientists are always at least open in principle to the idea that new evidence may come along that gives us a deeper understanding of what's actually going on. Now, so all that to say is that evolution is one of those scientific theories. It's a well-tested explanatory framework. It makes accurate predictions about the natural world. We've attempted to falsify it for about the last 150 years, and we have not yet been successful in doing so. So that it's a theory in the scientific sense. Now, evolution also, lucky us biologists, right, John? Evolution is also the theory in science that is most commonly a cause for concern among Christians for theological reasons. We, like me, we kind of grow up with that sort of idea that evolution and God are somehow in conflict with each other. Now, evolution happens to support two particular convictions that we'll touch on tonight that are often thought to be at odds with Christianity. Namely, that humans are the product of an evolutionary process. So this is not only a theory that applies to other forms of life on the planet, but also applies to humans as well. And that, secondly, not only that humans evolved, and this is more recent and sort of more recently on the evangelical radar, that humans evolved as a population. That at no time in our evolutionary history is there any evidence for a population that numbers lower than about 10,000 individuals. And I think, as I said in chapel or in the forum earlier, if you're like me, when you were in Sunday school, your flannel graph didn't have 10,000 people on it, right? It had 2. So that seems to be a little bit at odds. So what might one do with that particular info? Let's take a step back a little bit and sort of address this first question. If I tell you that evolution is a broad explanatory framework that has not yet been falsified and makes accurate predictions about the world, well, let's back up a little bit. There was a time when evolution was a hypothesis. There's a very famous diagram in one of Darwin's notebooks where he does a little sketch of a branching tree of relatedness, and underneath it he writes famous words where he says, "I think." That's a hypothesis. It's a way of thinking about the world, and it makes testable predictions. The hypothesis that Darwin was proposing was that species share common ancestors, and that things that we see in the present day might share common ancestral populations in the past. The idea here is that populations might become genetically separated, and then through genetic changes in those populations, new mutations, recombination, if those two populations are separate from one another, they're not averaging their new genetic changes across those two populations. So this means that differences can accumulate, average characteristics of the population may change, and those differences, if they're substantial enough and go on for long enough, may accumulate to the point where those two populations no longer recognize each other as members of the same species. Now, we're not that used to thinking about those kinds of average characteristics of populations shifting over time. We're not really geared that well to think about that as sort of just regular people. We're not used to thinking about things happening over long timescales like that. So this is an analogy that I develop in the book that a lot of readers have found helpful. How species change over time is very similar to how languages change over time. And all of us are at least a little more intuitively familiar with how languages shift over time. So similarly to species, we can have populations that become separated. This process of incremental change over time is also a feature of languages as well as species. So new syntax, new words, new changes in grammar and so on. This will occur in these different populations, and if they're separate from one another, those differences will not be shared. So again, differences may accumulate, average characteristics may change, and if you let this go on long enough, you may end up with new languages over time. So one way to think about this is that both species and languages are information systems, but they're copied imperfectly. So they have this ability to undergo incremental change at the population level over time. Now, we're all sort of familiar with this, especially if we're from the Christian tradition, because we're used to reading texts, say the Bible, from different periods in history, and we see the changes that take place. So I'm always afraid that when I do this talk in academic settings that I'll say, nobody here can read this. And there'll be that one prof at the back that says, actually, right? I can read it just fine. This is English. Well, it's actually Anglo-Saxon from about the year 990. And this is actually a verse from the Bible. This is John 1:29 from the Anglo-Saxon Gospels. And now all of us that were memorizing verses in Sunday school, we're spinning our little scriptural Rolodex to try to remember what John 1:29 is, right? If we look at it in a modern translation like the NIV, it reads very naturally for us. The next day, John saw Jesus coming toward him and said, "Look, the Lamb of God who takes away the sin of the world." One of the reasons why I like this as an example is it actually has Middle Earth in it, which is just a little extra value for the Tolkien fans in the crowd, right? The doing away, here is the doing away of Middle Earth's sin. Now, only in some sort of abstract academic sense is this the same language as this. But we know that these are two discrete samples pulled from what, in fact, is a continuous process. This is the same population of speakers speaking the same language generation by generation, but at the end of the day, we get this, whereas this is our starting point. Now, we can easily fill in some of the gaps because we have translations of the New Testament from different time periods, and I know you're going to kind of be squinting here. But the idea is that this is sort of like a fossil record, right, of this particular verse. Now, it's not the case that everybody sat down in 1394 and said, we've been saying it this way for a couple hundred years. It's time to change. Let's get together and get a new, This is an incremental process of continuous change over time from which we are drawing discrete samples. And that's a very good analogy for how biological species change over time, too. Each generation speaks the same language. Grandparents, you speak the same language as your grandchildren, mostly, right? Not perfectly. There's incremental change over time. And those differences can add up. So this shift of average characteristics of a population over time means that separate populations might become mutually unintelligible to each other, or they might become new species. But the process is a gradient, and it's decidedly difficult to draw lines of demarcation on a gradient, right? Humans like to categorize things into discrete categories, but gradients are hard to draw demarcation lines on. Now, it's actually very easy to go around and identify languages that are closely related to each other. Similarly for species, what we simply look for are shared features that exceed what is necessary for the system to function. That's a sort of biological way to put it, but you get the analogy in languages. With a last name like Venema or Venema, you would know that I have Frisian ancestors. Modern-day West Frisian happens to be very closely related to modern-day English, but they're mutually unintelligible to each other. Here's a sentence. This is a very famous example that shows the correspondences between West Frisian, modern West Frisian, and modern English. Butter, bread, and green cheese is good English and good Fries. Now, that is pronounced almost identically in Frisian as well. Now, there's no need for that amount of correspondence between these two languages. There's nothing intrinsic about the word green that is connected to that phenomenon, that particular wavelength of light. So why do we see these correspondences over and over and over again? Well, the speakers of modern-day English and modern-day West Frisian shared an ancestral population of speakers that lived on the European continent in the Friesland area at about the year 400. So Old Frisian and Old English are even more closely related to each other. These two populations were separated. Some of those folks went and settled on the British Isles. Of change in their two languages, and over the ensuing hundreds of years, their languages became slightly different, although they are among the most closely related languages today, but mutually unintelligible to each other. My grandparents spoke a wonderful mix of, my dad's parents, of Fries and English and Dutch, because they knew all three languages, and they just picked whatever was the best word to, so it was this beautiful mix. But even as a kid, I can remember hearing words and thinking, that sounds a lot like an English word. Why is that the case? Well, they have a history. They have a shared history. So similarly, we can look at the same sorts of things in biological species. Look for correspondences that don't necessarily need to be there. Shared similarities that might be hints that there's a common ancestral population in history connecting those two modern-day species. I don't have time to give you this evidence, but if you sort of take it on faith for a moment, that the evidence that we have from genetics very strongly supports the hypothesis that present-day whales, this is a group of organisms, whales, dolphins, and porpoises, is a group of organisms called cetaceans, aquatic mammals. The genetic evidence very strongly supports the hypothesis that they share an ancient common ancestral population with terrestrial four-limbed mammals. Darwin actually proposed this because he was aware if whales are mammals, mammals should be four-limbed. Can you think of any other mammals that don't have four limbs aside from whales? Right? Whales have front flippers and a tail. They don't have hind limbs, but every other mammal does. Now, whales are mammals. They have all the other mammalian characteristics. So evolution was kind of backed into a corner here. The alternative hypothesis is that this independent lineage just happened to independently come up with all the same innovations that characterize mammalian life. And that was seen as too far-fetched. So Darwin was kind of backed into this corner. Yes, whales should be the descendants of four-limbed terrestrial mammals. He got quite a bit of mockery for that suggestion. He published it in the Original Origin of Species. This is a gentleman, Mr. Seeley, writing sort of a review of Darwin. He says, "Thus, Mr. Darwin, he finds it impossible to believe the plain words of Moses that on the fifth day God created whales." Now, wait a minute. Anyway, we'll let that one go, but those of you that know Genesis 1 know that maybe there's the words there about God creating whales aren't quite so plain. "Sees no difficulty in believing that a race of bears." Now, Darwin was sort of hypothesizing what sort of race of animals, what sort of variety of animals might have led to the whale lineage. And he was thinking about the odontocete whales, the toothed whales, and thinking, okay, this is going to be a carnivorous ancestor, so maybe something like bears. And he had actually had correspondence from a biologist in North America who had seen a bear swimming and catching insects in the water, which is not that common for a bear to do, but that was what he had observed. So Darwin was kind of speculating, maybe something like that, given the right conditions over time, might have become a whale. He says, by contracting a habit of swimming, gradually lost their legs and were developed or evolved, developed and evolved, that those words are very close at the time in English, into whales 100 times their own bulk. And this sort of trash is called science. Now, Darwin was a man ahead of his time. Interestingly enough, Darwin was so embarrassed by this particular incident that he actually withdrew a lot of his text about whales in the next edition of The Origin because it was seen as just too outlandish. Now, I've told you that whales should be tetrapods. They should. Present-day cetaceans actually do have four limbs, but only for a very brief portion of their embryological development. So mammals, as a rule, start their development in very characteristic ways. And one of the things that they do at a certain time in development, they make what are called forelimb buds, and they make what are called hind limb buds. And whales, like any good tetrapod, make forelimb buds and hind limb buds at the same time that other mammals do. Interestingly enough, later on in development, not too much later, there's a second developmental program that is unique to cetaceans, which only operates in the hind limb area, which is then turned on and shuts down the activity of the other developmental program that is attempting to make a limb. So this is sort of cobbled on to shut it down after the fact. So early in embryological development, whales and other cetaceans have hind limb buds like any mammal should have, but these are lost later in development as this other program halts and then reverses that development. So these will regress back into the body wall. Another good feature of mammals is that they have two nostrils on the front of their face. That's sort of a defining feature of mammalian life. Whales, of course, have a blowhole on the top of their head. But embryologically, we also see that whales, like any good mammal, start with two nostrils on the front of their face, just like any good mammal should. And it's only later in their embryological development that these will migrate. You can see the migration is sort of partway done here. This will end up on the top of the head later on in embryological development. But they have the characteristic mammalian features, but just for a brief portion of their development. So we have not yet been able to reject the hypothesis that whales are modified descendants and share ancestors with terrestrial tetrapod mammals. The fossil record of whales was not well known at Darwin's time. Mr. Seeley, I mentioned earlier, one of the things he does in his review, he says, we can tell that this is just outlandish because we can look in the earth and we see bears and we see whales and we see nothing that would suggest any sort of intermediary form between them. It took scientists a long time to figure out where on the planet this transition may have taken place. It actually turns out to be present-day Afghanistan and Pakistan is where this took place. Great place for paleontology, except for all the political unrest. But once we figured out that this was the place to look, we very quickly found a number of species that blur the distinction between present-day whales and small terrestrial four-limbed mammals. Not that any of these are direct ancestors of present-day whales. They're cousins, sort of. They're relatives. It's not necessarily the direct lineage, but these are relatives that show us what sort of features were present. Interestingly enough, this one species here, the basilosaurids, they were known at Darwin's time, and you're going to have very, very sharp eyes, but you'll notice that they actually have tiny, tiny hind limbs. This is an aquatic, fully aquatic organism. These tiny hind limbs are not capable of supporting the bulk of this large aquatic animal. The fossils that were known from this group of species at the time of Darwin were not well preserved enough to preserve those small hind limbs because they're actually not connected to the skeleton, to the rest of the skeleton. They're not articulated with the rest of the skeleton. They're only connected through muscle. So in that sense, they're not functional in that sense as a leg. Now, if that had been known at the time of Darwin, I wonder if Mr. Seeley might have had, been given a bit of pause before tearing into Darwin in the way he did. So that's some embryological evidence, a little bit of fossil evidence of what biologists see. Of course, in the present day, most biologists, when it comes to evidence for evolution, are well aware that most of that evidence, or a large amount of that evidence, came through genetic sequencing. I sometimes tell my students, and I told a group of students here earlier today, that if we had never come up with this idea of common ancestry, if Darwin had never lived, if Darwin and Wallace hadn't come up with this idea, by the time we started doing genome sequencing and comparing the sequences of different organisms to one another, we would have been forced to that hypothesis in very short order, just by looking at what we were seeing, right? So one example of that are these things called unitary pseudogenes. It's a historical name for false gene. When they were first discovered by biologists, they could tell that they had certain properties of genes, but not all of the properties. So they were a little bit enigmatic at the beginning. As an analogy, if you were walking across a field and came upon this, you would know some things about it. You would know it's a building, but you would also know it's not in its original state of repair. The roof is missing, there's grass growing up through the floor. So you would rightly conclude, unless you've actually wandered accidentally onto a movie set, that this was not deliberately constructed in this original form. It had a certain form and function prior, and then that form and function has been partially lost. But it's still very much obvious that a building is here. It's just not in full repair. The same sorts of principles apply to genetic sequences. Biologists can look at sequences that are gene sequences. They should have certain features in order to be functional gene sequences. They have what's called a promoter, and then a sequence that codes for the protein that they will make, and regulatory DNA sequences that control when and where it is made. It's possible that mutations can come into those different sequences that stop that gene from having its original function. The interesting thing at that point, though, is that just like buildings in a field, that structure can persist in a lineage for a very long period of time. And what's going on there is that the enzymes that do DNA copying, they don't know the function of what they're copying. They simply try to make as faithful a copy as they can. Now, if they make a mistake, which is where these mutations come from, the next time it's copied, they don't know that they've made a mistake previously, so they don't fix it. They simply try to faithfully copy it. So because of that, it can persist in a lineage for a very long period of time, even after it has lost its original function. One of the things that immediately struck biologists when they started comparing sequences of multiple species is that not only do we see mutations in many genes, lots of these pseudogenes present, we often see the same mutation in the same gene shared across multiple species. So here's an example of that. These are genes that are dedicated to the olfactory sense, the sense of smell. This column here just tells you what the gene name and location is in the genome, and then we're going to compare humans, chimps, gorillas, and orangutans. Wherever you see text in the column, that indicates to you that the gene is not functional, that particular gene is not functional in that organism, and it's telling you exactly what the mutation is. So say one down here, this gene in humans has a deletion mutation at DNA letter position 178 in that gene. But then again, you'll notice that that same mutation down to the exact DNA letter is also present in chimpanzees, gorillas, and orangutans. So there's a couple of hypotheses that you could use to explain that pattern. One hypothesis is that that exact same mutation, down to the individual DNA letter, happened independently in 4 unrelated lineages. That's one hypothesis. The other hypothesis is that this mutation happened once in an ancestral species, and then that particular genetic defect was inherited by a number of species that descend from that particular common ancestor. And that explanation, of course, is a lot more reasonable because it requires very small number of rare events. The independent mutations multiply happening in separate species is not something that biologists think is probable, and we've shown that it's not very probable. If you take this data set, you'll notice that there are some things that are shared, and then you'll notice that there are some things that are unique. Lineages might have unique mutations as well that are only in that particular species. If you take this data set, this small data set from this one paper, it falls into this particular pattern. So we see some mutations that are identical to a species, and then we see some that are shared. So here are 3 that are shared between humans and chimps. Here's 3 that are shared between humans, chimps, and gorillas. Here's 6 that are shared by all 4. There's also something else to notice about the pattern. For example, if we see a particular mutation in humans and it's also present in gorillas, we can predict with great confidence that that exact mutation should also be present in the chimpanzee genome, because the common ancestral population of the human lineage and the gorilla lineage also gives rise to the chimpanzee lineage. And we do, we see that pattern. Likewise, if something is present in humans and in orangutans, we will also expect it in gorillas and in chimpanzees, and we observe that. So this is what's called a nested hierarchy. It's kind of like Russian dolls, right? If you think of species as kind of nested Russian dolls, whatever happens when they're nested together gets propagated even when they get pulled apart and separated. So there's nothing out of place, at least in this small data set. I really enjoy these kind of examples because it's amazing to me how long a pseudogene can actually persist in a lineage. This is another study that I like to use as an example, and I talk about it in the book. There's a lot of good evidence to suggest that placental mammalian biology is a modified descendant of egg laying. I don't have time to give you all the other lines of evidence, but that's what it would suggest, that mammalian biology, including placental mammalian biology, is an innovation within a group of egg-laying organisms. You might be familiar with, there are still 2 lineages extant on the planet of egg-laying mammals, the platypus and the echidna in Australia. So there are egg-laying mammals, there are marsupial mammals, and then there are placental mammals. And those are thought to be innovations within an egg-laying group. So this research group was interested in this question, and they said, okay, there are certain genes that are devoted to egg yolk production, and they're identifiable. These group of genes called vitellogenins. So they had this idea, and this has got to be some grad students just sort of sitting around one day and saying, yeah, I mean, you can just do this by looking on databases now, right? So we call it doing experiments instead of in vivo or in vitro. It's now in silico, in computer, right? So don't tell the PI, but I bet you we could find the remnants of vitellogenin gene sequences in placental mammals. Let's go look. Well, how would you go about with doing that? Okay, let me tell you a little bit about vitellogenins first. They're a protein that's required for egg yolk formation in birds and other amniotic organisms. Placental mammals are thought to share a common ancestor with modern-day amniotic organisms like birds approximately 310 million years ago. Placental mammals maintain a connection with our embryos all the way through development, through the umbilical cord and the placenta. An egg-laying organism faces a particular challenge. Once that eggshell is deposited, that breaks the connection between mother and the embryo. So the way amniotic organisms get around that is they provide nutrition in the form of the yolk. And vitellogenins are a big part of that. They're a protein that's a really large protein. It's got lots of sugars stuck all over it, and it's also, its 3-dimensional structure is very attractive to lipids. So it's kind of this nice 3-in-1 nutrition package. They're made in the liver of the mother, they're transported through the bloodstream, they're deposited in the egg yolk. Most of what you see when you crack a chicken egg and you're looking at that yellow, a lot of that is vitellogenins. And it's intended to be nutrition for the developing embryo. It's not meant to be your scrambled eggs in the morning, but it's good, right? So how did they go about trying to address this question? I know, I just ruined breakfast for you for I'm sorry. The reason why it tastes so good is because it's, sugars, amino acids, and lipids all in one. So they looked in a present-day amniotic organism, in this case chickens, and a present-day placental mammal, and it just so happens that humans are now the best described placental mammal with respect to their genome, because we've spent so much time and effort researching the human genome. So what they did is they simply looked to see where these genes are in the chicken, and there's 3 of them. They actually happen to be evolutionary duplicates of each other, but that's another story. There's other genes nearby in the chicken, this thing here, this thing here, this thing here, and this thing here. These are genes that are not part of egg yolk formation. They just have other roles. Those genes are also present in humans, and they do the same role in humans that they do in chickens. Because, there's standard biology, and these are part of standard biology that's common to both humans and chickens. Now, the hypothesis that we're testing is that 310 million years ago, that this stretch of DNA and this stretch of DNA in a present-day human and a present-day chicken were once the same stretch of DNA in the same ancestral species in common, and they've now gone their separate ways for 310 million years. So what this research group did was very straightforward. They're going to simply look in this region, and in this region, and in this region, to see if there are any sequences here that show a correspondence to the present-day sequences that we see in chickens for these genes. And these little black bars are the result of that test. Wherever you see a little black bar, that is a sequence match between the human genome and the chicken genome. And you'll notice that the matches spread across these regions, giving us confidence that, yes, we're looking at heavily modified descendants of the original region and the original ancestral species. And most importantly, for our purposes, we see small fragmentary sections that have sequence matches with present-day vitellogenin genes in chickens. So if you use evolution to make a prediction about where you might find the remnants of genes devoted to egg yolk production in a placental mammal, that hypothesis is supported. We cannot, based on this evidence, reject the hypothesis that humans and chickens shared a common ancestor and that these 2 sequences were once the same sequence in a common ancestral species. Sorry for making you feel guilty next time you crack an egg, right? Okay. Here's another one, just to sort of drive this point home a little bit further. This is another paper. I won't spend a huge amount of time on it. This is a particular gene that is known to be a pseudogene in the human lineage. It, too, has a number of other genes around it, just like you would expect it. This is a map that's actually more of an accurate map as opposed to sort of the cartoon I was showing you before, although that previous cartoon is drawn to scale, in case you're wondering. Here's what it looks like in the mouse genome. So you'll notice that this same gene, which is a pseudogene in humans, is also present in the mouse genome, and it is a functional gene in the mouse genome. And we actually can understand and experimentally discover and have discovered its function in the mouse. Now, it's a little bit tricky because there's been what's called a chromosome inversion here, but you'll notice this corresponds here, and so on. It's like we took this whole region and just flipped it, and now it's in this orientation in the human. That sort of thing can happen, chromosome breakage, rejoining, especially since mouse and human are very widely diverged. We haven't shared a common ancestor for quite a long time. Now, what's interesting here is that, okay, we see it as a functional gene in mouse, we see it as a mutated gene in humans, we can understand the function in the mouse and test it. We know that this gene does not have that function in the human genome, but there it sits. And just because biologists get bored, I decided to say, okay, if that pseudogene is present in humans, that very strongly predicts that would also be present in the chimpanzee genome, because humans and chimps share a much closer recent common ancestor than we do with mice. And my prediction before I did this little analysis was that that mutation that took out that gene in the human lineage, that mutation occurred long before humans and chimps went their separate ways. So this is just publicly available databases. This is that gene that we were just looking at. HS is Homo sapiens, so that's the defective human gene. Here's the functional mouse gene. This happens to be coding sequence, so the biologists in the room will recognize that familiar ATG. This is where the protein should be starting. And you'll notice that down here, we're missing a DNA letter. This would have catastrophic consequences for this protein. It would not be able to be translated properly. And lo and behold, that exact same mutation is also present in the chimpanzee genome. supporting my hypothesis before I went and just pulled this out of a database, that this mutation, the mutations that inactivated this gene occurred before humans and chimps went their separate ways. But we see this gene in mice in the same context, doing a job. It's there in humans, but defective. It's also there in chimps with the exact same mutations that remove its function. Okay. So that hopefully at least gives you a bit of a flavor for why evolution, or why biologists are so convinced that evolution is a predictive framework. We can use it to make all sorts of predictions about what we should find in genomes. And this is just a tiny slice of what's out there. It remains, 150 years on, an extremely productive framework for understanding the facts as we have them. Now, so that's sort of the first point, evidence showing you that humans and other forms of life do share common ancestors, that evidence is quite strong. The problems don't stop there, unfortunately. Not only do we descend through an evolutionary process, as the evidence strongly supports, I've also told you that we did so as a population. You know, it's sort of like back to our language analogy. We don't normally think that new languages start when two people go off and start speaking a different language, right? We at least have that kind of feeling that that's not normally how languages start. That's not normally how species start either. You have populations that separate, begin to accumulate, change, that sort of thing. So our first expectation is probably not that the human species started with just two individuals from a biological perspective. There's actually no evidence in our history for a population, like I mentioned before, that numbers less than about 10,000. There's a lot of independent ways to estimate ancestral population sizes. We're doing that from looking at current genetic diversity, understanding the principles that generate that diversity, and then extrapolating back in time to ask the question, well, how many ancestors do we need to give us the diversity that we have in the present day? Now, all of these methods, of course, like any method in science, they have their limitations. Interestingly enough, all the different methods we've thrown at this, even though they have different starting assumptions and make different, are based on different principles, they all agree that we descend from this population of around 10,000. I'll show you one example. This gets us into the weeds a little bit, so if you kind of glaze over for this and this kind of all sounds like gobbledygook, just come back in 5 minutes and we'll be good, okay? So one method that tests this can look at the DNA variants that are present in one person. So we all have one copy of our sequences that we got from mom, one copy we got from dad. Now, those two sequences may not be identical to each other. They may have differences. Although if you track back far enough in human lineages, eventually you're going to get to a point where those two sequences are the same. So the number of differences we see in the present day is now an indicator of how long those two sequences have been separate from each other and how many mutations they've accumulated since they were the same starting point. So that's called coalescence, or the time to the most recent common ancestor. So we're looking at different variants within a gene. How long has it been since those two variants came from a common ancestral source? Okay, so if we have two variants, they have DNA differences. What we're sort of doing is mentally working backwards and saying we see differences in the present day. Let's mentally work backwards through the different mutation events that made them different until they coalesce and they're the same entity that then becomes later different through mutation. So coalescence happens at the time that they share a common ancestor, so the time to the most recent common ancestor point. Now, the interesting thing is the time to the most recent common ancestor is actually proportional to the size of the population. So basically what's happening, when we look at data, this is a simulated data set, but the same thing applies to present-day chromosomes. We're looking and saying, how long has it been for the time to the most recent common ancestor? For sequences that are very, very similar to each other, the time to most recent common ancestor will be very short, right? If you in your family history, as most of us do who are Northern Europeans, if you're Northern European like me, we would have relatives, like as we go back in our family trees, and if you've done your pedigrees and that sort of thing online, it's very common, say 4 or 5 generations back, that you might find an ancestor that you inherit from on both sides. So if you have a sequence that you've inherited from that one individual down both sides, then the time to most recent common ancestor is just a couple of generations ago. And that sequence would be very, very similar because it probably hasn't picked up any or very few mutations in it. On the other hand, some of your DNA, some of your chromosome segments might have quite a few differences between the two copies. So in order to explain that, we would have to reach farther back in time. It's been much longer since those sequences shared a common ancestor. So you might see this sort of spike where you suddenly have a lot of difference there. Now, let's think about population size. If you have a really small population that you descend from, then the probability of coalescence or sharing a common ancestor from both sides gets higher, right? This is actually a feature in my wife's family because my wife, she's very interested in these genealogical questions, and she's found out that she's related to about 14 or 15 people on the original Mayflower that came over. Now, that's not that uncommon. There's a few million people in the United States and North America that have that. But if you're related to one, you're most likely related to a bunch of them because it's a small community, and they intermarried and had kids together in that small community shortly after coming to North America. So for those regions of her genome, she would have very short coalescence times because of that small population. The smaller the population, the more likely it is you're going to have a coalescence time. Now, imagine a severe bottleneck where you have a very small number of individuals in the breeding population. It's quite probable in that scenario that some genes are going to lose all variants except one. There's only one variant that's going to pass through that bottleneck because of that small population size. So that is going to force a time to most recent common ancestor for that particular variant, for that population. So what we can do with an individual is we can simply look along their genome, compare the different variants that we see as we go along a chromosome, calculate how long the divergence time is, what's the time to most recent common ancestor as we kind of go step by step along the genome. We can take those values and we can plot them as a distribution. When we plot them as a distribution, we might notice some peaks in that distribution. We might say, wow, at 10,000 years ago, we suddenly have this large spike. There's a lot of coalescence going on at 10,000 years ago. Or there's a lot of coalescence going on at 2,000 years ago. Well, that is a suggestion that at those time points that the population size was small because the probability of coalescence was high. So it becomes a way to extrapolate into the past and ask this question, how large was the population based on what we see in current day genetic diversity? So all of that just to show you one slide, right? Don't you love science? So this is real data now for real human populations. This is the effective population size in tens of thousands. But really what's going on here is just probability of coalescence at different time points. And this is time into the past. It's a logarithmic scale. So here we have 10,000 years ago, 100,000 years ago, a million years ago. And what we see for different human populations, here are two that are sub-Saharan African populations. And you'll notice that, yes, they have a dip in this time frame. So this would be, about 50,000 years ago-ish. You'll notice that there's a dip, but it's a dip that's still to several thousand individuals. So sub-Saharan African populations are maintaining an effective population size by this analysis on the order of several thousand. If you're not from a sub-Saharan African population, that's because the rest of the planet was populated by a small group of individuals who migrated out of Africa right around this time point at about 100,000 to 50,000 years ago that then went on to populate the rest of the planet. And so those of us who are not sub-Saharan African, we descend from this smaller population of individuals. Those individuals, by this analysis, we can see dip down to a population size on the order of about 1,200-ish or so. But taken together, this comes up to about that 10,000 figure. And you'll notice at no point in the time frame that's represented here do we see anything that sort of gets close to what we would think of as 2. Okay. Here's another method. This is even more technical, so I won't spend a huge amount of time on it. But it's a nice figure because it shows the distribution that we have of the time to most recent common ancestor in our genomes. This is also what's called a cumulative distribution function, so it's a little bit hard to read, but basically we go from 0% of the genome to 100% of the genome, and we ask, what does it plot in terms of the number of generations since there was a shared common ancestor? And you can see there's different kinds of DNA that are being analyzed here. I'll focus your attention on the neutral DNA because that's the most reliable for our purposes. And you'll see that there's this distribution, and this corresponds with us being comfortable with the statement that we can't, if we wanted to really sort of push this analysis and ask the question, could we have a population that's down to 2, which would mean that they would only have a possible maximum of 4 variants at any particular location in the genome? How long ago would that have to be based on current genetic diversity? And that's really pushing this to the limit. And the answer is right around 500,000 years ago, where that would possibly be sort of just inside the error bar for this kind of analysis. Okay. That's what's called the time to the most recent 4 alleles or 4 variants. Okay. Now, that's genomics. Genomics is its own field, it's its own discipline, it has its own rules that it plays by. It's very well suited to asking certain kinds of questions, but Christians obviously have other kinds of questions that come out of data like this. And science, of course, is not the best tool to answer those questions, right? So genomics evidence is really well suited to addressing material origins, where we come from, what that mechanism might have been, but it's not well suited to address other questions that are strongly of interest to evangelical Christians, like, are Adam and Eve historical individuals? What I've shown you is that the genomics evidence very strongly supports the hypothesis that we've never been a small population throughout our trajectory, but it doesn't say anything about the particular historicity of a person named Adam and a person named Eve. What it weighs against is sole genetic progenitorship from those individuals, from 2 people. But the historicity of somebody named Adam, somebody named Eve, science isn't really well suited to address that particular question. You know, how and when did sin enter the picture? Well, give me, as the geneticist, a tool for detecting the genetic signature of sin, and maybe I can give you some idea. Right? Science can't answer that question, right? But it's a question that we're curious about. How might the church integrate this genomics evidence into its worldview? These kinds of questions and other ones that are related to them, for this kind of question, the scientists have to be humble and sort of hand over the baton at this point and say, to the best of my ability, as I have sought out the wonders of God's creation, this is what I have found. And then the theologians on the other side say, thank you for that, and now, you've created all these problems for us, and now we have to do some work, right? That kind of thing. I jokingly said to Scott that my job in the book is to create all the problems, and then his half of the book, good luck, you got to solve them all, right? Whether or not he succeeded, I guess, remains to be seen. Now, keep in mind that science might call us to return to the text with renewed focus, and this is not new. Science has called Christianity to return to the text multiple times. Now, that does not mean that that science somehow determines what our interpretation of the text might be. Science doesn't have that power. Science at best can flag up for us that maybe we have misread the texts because we have unknowingly brought in our Western scientific worldview, perhaps, to our reading of Scripture. So it can flag up that there might be a problem, but it's not going to tell us or dictate to us how we might interpret the text. The texts need to be interpreted by the tools and techniques that the theologians and biblical scholars bring to bear. So it might flag up that there's a problem for us, but it's not going to predetermine what those answers might be. Let me ask you this question. If you're like me, like I said at the beginning, you might have grown up in an environment where evolution and creation, evolution and design, God's intelligent design, his plan, that those might have been presented as polar opposites, that they were somehow incompatible with each other. A thought that I would like to put forward to you is that possibly that is a category mistake. What if it's possible to view evolution as a form of design, as God's intended mechanism for bringing about biodiversity? Now, why might I think something along those lines? So these are just a few examples of things that I'm thinking on now. I am working on a second book that is tentatively titled Evolution as Design. the idea that evolution could be viewed as God's intended mechanism for bringing about biodiversity. Interestingly enough, human engineers are starting to use evolutionary methods, and they're using them to solve design problems that are previously intractable, things that are difficult to solve. So the way this works, things that are too complex for standard methods, you can do this sort of thing. You can design code that can replicate, self-replicating code. And you can design it with mutation, varying mutation rates within that code, so there's variation. Then you can randomly test the mutated descendants for a desired function, and then you can repeat this process in an iterative manner until a solution is arrived at. Now, the interesting thing is that good solutions are found by this method, but not necessarily optimal solutions, and that's actually what evolution does. It doesn't necessarily produce optimal outcomes, it produces good outcomes. That sounds a little abstract. Let me show you an example of how this has been done. This is some work that has been done by scientists working for NASA. So the goal here is to develop antennas for use on spacecraft that have particular characteristics and are resistant to certain types of feedback and other things that I don't understand because I'm not a physicist, right? But you've got to have antennas that perform within certain parameters for certain missions, for certain satellites doing different functions. Now, there are traditional ways to do antenna design. They're very labor intensive, and this group was interested in trying to apply an evolutionary type of algorithm to try to design these antennas using a way that they couldn't necessarily predict. So they actually went through the trouble of writing code that would produce different shaped wires that then they could assess for function, and then they would pick the best ones out of the first batch and then do another iteration where they would make more, and so on and so on. And they ended up coming up with these designs. Now, these designs work extremely well, and they actually outperform the designs that were sort of designed the old-fashioned way. The length of time that it took for this process was extremely rapid compared to the usual methods. And it produced some pretty odd-looking designs. Here's one design where it's actually branching, and there's, I think, 4 different branches that are all identical. So they evolve each of these independently, and then they just make 4 duplicates of it. That was the first experiment. Interestingly enough, then the mission parameters changed, right? The government said, there's, I don't know, funding changed or whatever. And suddenly they have to do a different design on short notice. So they said, okay, let's not do the branching 4 repetitive model. Let's just do 1 wire. And they ended up with this. And this performed beautifully, even though it's nothing like what they would have sort of intuitively come to as part of their design. And they got it ready in virtually no time at all. So they say, current practice of designing antennas by hand is severely limited. It's time and labor intensive. You need to know a lot before you start. But evolutionary algorithms could be used to search the design space and automatically find antenna designs that are more effective than would otherwise be developed. So, and they actually flew this thing on a satellite, and it worked really well. It was the first example of an evolved genetic algorithm or evolutionary algorithm evolved piece of hardware that flew in space and performed wonderfully for the mission. This is actually very similar to an example that's known within biology as well. You might know a little bit about the immune system and the antibodies that it produces in order to fight off pathogens. One of the things I teach my students when they're learning about immunology is that there's almost limitless pathogen pool out there. Humans have limited, any vertebrate, we have limited genetic resources to fight off these hordes. Even if we dedicated 1 gene per pathogen, we only have about 20,000 genes. So that's not going to cut it. So how do we go about solving this problem? And the way we solve the problem is by this sort of genetic algorithm type of evolutionary algorithm kind of process. So antibodies that bind really tightly to pathogens, to nasty stuff, this is how we go about making them. We take a portion of the antibody gene. Here's just a picture of what this antibody protein looks like, kind of in real life. We randomize a portion of it in its sequences in each individual antibody-producing cell. We select cells that have good binding and we say, okay, duplicate those. And then we actually do further mutating during that copying process. And again, we select those with even tighter binding. And we can do this in an iterative manner such that in a very short order, we will develop antibodies that have very, very high affinity for binding onto the pathogen target. Now, can we predict in advance what the features of that antibody will be, what its sequence might be? No, we can't predict that. But we can predict with very great confidence that something will be found that will be very, very effective. We can't predict it in advance. And the fact that this randomizing and selection process is part of the picture, this is what allows us to fight off this almost limitless pathogen horde with a very small number of genes, because it's got this randomization process as a part of it. Okay. So it's possible to think of evolution not as sort of a random, strictly random process, but a process that is a search for solutions. The type of thing that a designer might actually deliberately employ in order to solve a particular problem. Human designers are using these methods, and it stands to reason, at least to my thinking, that if human designers are capable of realizing the power and efficacy of these methods, that surely God, as a designer who is much wiser than we are, would have a much better understanding of how to use these principles to accomplish what he would like to accomplish in his purposes. I think given the time, I'll probably skim by this. It's fun, but I've thrown a lot of data at you. That's just good evidence to show that gene functions, new genes have been developed over time in evolutionary history, and those genes have been put to new functions. So evolution has this mechanism by which it randomizes and then out things that work. But we've already sort of seen that with other examples. So one Christian approach to evolution is to think that and to view evolution as a powerful mechanism that was designed by God, that he deliberately used to develop biodiversity on Earth. Of course, this goes beyond what science can tell us, right? Science can tell us evolution has these properties, it has these features. I can then take off my scientist hat a little bit and step to the side and say, wow, that's amazing that it has those features. It doesn't surprise me that God would use that as a mechanism for doing his work. Now, science can't take me to that point, but science can't take me to that point for other disciplines either. If we think about things like Mendelian inheritance or developmental biology, we're generally pretty comfortable with viewing these as natural processes that God has put in place, designed by him to accomplish the purposes that he intends. When I teach Mendelian inheritance or developmental biology, I have not yet, in my 15 years at Trinity, had a student object and say, Dr. Venema, you've told me all these wonderful things about how organisms develop and the intricate processes that do that, but you're leaving God out of the equation, right? Or my colleagues in physics, as they teach Newtonian mechanics and forces and those sorts of things, those laws of motion, they have not, to my knowledge, had a student say, but professor, where's the part that God does, right? We're comfortable as Christians with understanding that God can work at his choosing through what we would call natural mechanisms, regular patterns that we can explore using science. And we're also comfortable with the notion that God at times may choose to use supernatural processes to accomplish his purposes. It's God's show, he can choose what he would like to do. And in principle, Christians are okay with either option. I think where the challenge sometimes comes for us is when we're used to thinking of it in one category, so how species come into being, how God made species. We were used to often thinking of that as a supernatural process. And now we're shifting from that category to maybe say, oh, maybe God chose to use what we can describe as a natural process for that particular part of creation. We're okay with that, I mean, for Newtonian mechanics, although at the time, there were Christians who objected to it because it seemed to be removing something from the domain of God. Now, that logic only works if you think natural explanations and supernatural explanations are somehow a zero-sum game, that everything you explain by science is one less thing for God to do. Well, that is not and never has been a Christian position, right? Christians have long said that God is the author of nature and the author and a supernatural agent, and that he is able and does use both natural mechanisms and supernatural mechanisms to accomplish his purposes. So if you don't have problems with these things, hopefully you will eventually not have problems with evolution. Okay, I would argue that a truly Christian approach has to take both Scripture and science seriously. We can't ignore either. We shouldn't uncritically accept either as well, right? But these are both sources of God's revelation because he is the author of both. God is the author of creation, the creator, so what we see in general revelation should match up with what we see in special revelation. So at the ultimate end of the day, there should be no conflict between those two things. Now, of course, we appropriate special revelation and general revelation through human methods, exegesis and science, for example. These are fallible human activities, so it's possible that there may be apparent conflict even if there is not, in reality, conflict between these things. Now, there's a range of positions taken on Adam and Eve, even among Christians who accept the evidence that I've shown you for evolution and population sizes. Some positions affirm that Adam and Eve are historical individuals who acted as representatives of a larger population. Individuals along these lines would be N.T. Wright or John Walton. Other positions view the Genesis narratives as symbolic stories that are written in a genre that's familiar to the ancient Near Eastern recipients of the original text. So along this line of thinking, if you could sit an ancient Near Eastern Jew down for coffee and ask them, what do you get out of Genesis 1, 2, and 3? What they say would be markedly different than what we would think is supposed to be coming out of the text, because we're reading it potentially through sort of this Western scientific worldview, and they're taking things out of the text that we wouldn't necessarily twig to because it's not our culture. These things are not necessarily mutually exclusive as well. There's a lot of variation here. One of the things that I like to encourage my students with is that affirming a literal interpretation of Genesis has never been a requirement for orthodoxy within Christianity. There's always been variation. And if you look back at the patristic fathers, there's a lot more variation there than we would even be comfortable with in the present day. My colleague, Craig Allert, I'll give him a shout out here, just recently wrote a book called something along the lines of Ancient Interpretations of Genesis 1. It's all about the patristic fathers and their take on Genesis 1. Highly recommended. This is what Augustine says in his commentary, The Literal Meaning of Genesis. As an aside, Augustine's literal meaning of Genesis is that he thought creation happened instantaneously, and then God chose to write us a narrative to explain the theological importance of what took place. So literal interpretation by Augustine is sort of different what we would call a literal interpretation now. He says, in matters that are so obscure and far beyond our vision, we find in Holy Scripture passages which can be interpreted in very different ways without prejudice to the faith we have received. In such cases, we should not rush in headlong and so firmly take our stand on one side that, if further progress in the search for truth justly undermines this position, we too fall with it. That's from about 415 AD. So I would argue that a Christian approach requires humility. We have to recognize that both human exegesis and science are fallible human activities. But I would also argue that in cases where science or evidence remains strong despite many years of studies, decades or centuries, Christians, of course, have warrant to re-examine their interpretation of special revelation, because we're convinced that special revelation and general revelation have the same author. The role of a believing scientist in this conversation I see as one who provides the church with the best scientific information available, such that the theologians and biblical scholars can do their work with the best information available to them. It's also a missional issue for the church. Our, I'll argue, relatively infrequent or relatively recent inflexibility on origins issues in some quarters is a barrier to those outside of our community. If I want to share with one of my scientist colleagues the life-giving relationship of joining the family of God and having a relationship with Jesus Christ, and then I say, yeah, in order to experience that, here's a couple of things you're going to have to believe about science. It's a non-starter, right? There's another quote I shared with the students earlier from Augustine as well, saying, if we try to present something that that people know is at odds with their reason and experience, how can we hope that they will hear the message of the gospel? Paraphrasing here. So it can be a non-starter. So it's a missional issue for people outside the church that have scientific understanding. It can also be a barrier for those within the church who learn about this. Some of my students that struggle with this issue the most are ones that come from communities that have made, sort of as a defining feature of their community, a particular commitment to perhaps young earth creationism or something of that nature, where evolution is wrong. And for those students, then, it's not this question of just sort of this academic exercise of deciding where the evidence lies. It's questions about my identity. It's questions about my community. It's questions about what am I going to say to grandma when I go home for Thanksgiving, right? Like, it becomes this big issue. As an aside, students that come from traditions or communities that are more flexible and say, yeah, there's a range of Christian positions, they generally do fine. They're generally not stressed about it. So it can make a big difference for students. So it is a missional issue. I promised you earlier that you would just be getting the science half of the equation. Scott McKnight is the author of the second half of the book. His question is mostly, what do we do with the New Testament? What do we do with Paul? What do we do with Paul's use of Adam? And if you're interested in that, you can obviously buy the book and read his half of the book. Or there's a lecture that he gave. This is actually at my home church, South Langley Mennonite Brethren Church. He came up for a book launch, and we both gave presentations, and it was recorded, and it's online. So if you go to csca/Adam, that will direct you to Scott's half of the talk. I'll show a slide in a minute, but the CSCA is the Organization of Christians in Science in Canada, which is also a valuable resource for others. So CSCA/Adam will take you there. This is the CSCA information in general. Their website, csca.ca, and then, of course, /Adam for the talk. But there are many other resources available on this site and this is a Christian organization in Canada, an affiliation of those interested in science and scientists. Students, there is free membership available to you in this organization. It's an incredible resource, and students have the ability to become members of this organization for free. And I would highly recommend it. There's a journal that's available called Perspectives on Science and Christian Faith. I've written some articles for this journal over the years. Fantastic resource, highly recommended. csca.ca. Check it out. Other resources of note. I mentioned John Walton previously as an individual who holds to Adam and Eve as representatives of humanity. If you're interested in this, I would highly recommend these two books by John. John is an ancient Near Eastern scholar and professor of Old Testament at Wheaton College, and he's written these two books and others, but these ones in particular are very, very useful for trying to take our place in the audience, John would say, of the original recipients of Genesis 1 through 3, and trying to understand what they would have taken away from that material in its context. Highly recommended. Another resource that I would highly recommend, this is actually a sermon that was preached by my pastor. This is Dave Navarro, lead pastor at South Langley Church, South Langley Mennonite Brethren Church. Yep, I'm a Mennonite too, right? Dave preached a sermon a number of years ago. I know, I'm kind of just a recent convert to Mennonite life. I actually kind of got moved out of it at another denomination. It's a bit of a long story, but the Mennonites took me in, and I'm so grateful. I can remember having a conversation with my wife as we were kind of losing our fellowship with this other church, in part because of evolution issues, also in part because of women in leadership issues, but it's another story. And I said, either we're going to have to be the really conservative side of a really liberal church, or we're going to be the perceived liberal side of a more conservative church. We want our children to grow up in a community that believes that the Bible is true, that it's reliable, that Jesus Christ is a historical individual, that the resurrection is true, all of those things. I didn't want to go to a church that was kind of soft on those things. And I wondered if I would ever find a community that kind of would accept these ideas, at least as one of the faithful options, but wouldn't be wishy-washy on theology, right? Because I don't want to go to a church that's wishy-washy on theology. I'm actually quite conservative. I know it might shock you, but I'm actually quite conservative in my theology. I have found a wonderful home in the Mennonite Brethren Church. Dave wasn't the lead pastor at the time, but the pastor at the time, I took him aside on the second Sunday that we were there, and I said, sooner or later you're going to hear some things about me. And I'd rather you heard about them from me directly and not just from rumors. And I explained to him who I was and the type of work I did with the Biologos Foundation, which is an organization that promotes science within the church. And I said, I'm okay if you tell me that maybe we should just keep looking, and I don't want to disrupt your fellowship by bringing in controversy. And the pastor at the time, wonderful man, he said, you are welcome here. He said, there's a range of views here. Some of our people are young earth creationists, some are old earth creationists, some are evolutionary creationists like yourself. As long as you are willing to dialogue charitably and be in loving fellowship with all views, you're welcome here. And I said, absolutely, that is exactly what I want to do. And we've been so welcomed at that church. I just love it. Anyway, I'm going on here. Pastor Dave preached a fantastic sermon a couple of months ago in a series of messages called Text Message, and this one is about the reliability of the Bible. But if you want to hear a sermon from a pastor within the Mennonite Brethren tradition who loves God and loves Scripture and has done, in my opinion, an excellent job of balancing those things in a congregation, this is a sermon you should listen to. It's called The Reliability of the Bible. It was streamed on October 1st, 2017. You can find South Langley Church's YouTube channel on YouTube. Highly recommended. Wonderful. And of course, you can get the book if it's of interest to you too. My section of the book is similar to what I presented to you tonight, but other things as well. I also have a chapter in the book where I talk about intelligent design arguments and why I don't find them satisfying. I believe that God is the designer. I believe that he is intelligent. But the intelligent design movement, capital I, capital D, is a particular movement that I actually used to adhere to, but I don't anymore. If you're interested, the details are there in the book. And of course, the BioLogos Foundation, www.biologos.org. Fantastic resource. I wrote for them for many years. There's lots and lots of things on there about evolution that I can point you to, and writing by theologians as well. Fantastic resource, highly recommended. Okay, and with that, I'll thank you for your patience, and we'll open it up for questions and discussion. So thank you very much. If you have a question, there's a microphone here that you can come to. If you have mobility issues and you want to ask a question, just put up your hand and someone will bring you the microphone. If not, we'll just welcome you to the mic to ask your question. I would ask that it be a question. That's not always the case, right? And that it be reasonably brief so that we can have as many people as possible that want to ask a question. So I'll welcome any that have questions to come to the mic and ask at this time. If anyone's brave enough to be the first. Hello, sir.
Hello? There we go. Of everything you talked about, I think the part I found most interesting was on the idea of using evolution to research in other areas of science. Mm-hmm. Where can we get more information on that? That I found very interesting.
Yeah, there's actually, so the question is, using evolutionary algorithms in engineering and other areas of research, there's actually a whole body of literature out there. This is something that's been well used in many fields for over a decade now and has been very successful in many computer science approaches. So there's a whole literature. If you just do a Google search on evolutionary algorithms, that would definitely pull up reading material that you could get started with.
Thank you very much.
It's very cool. It's so cool. Other questions?
I'm Gord Zerby. I teach here in the area of Bible.
Okay.
Won't mention anything else, but I was interested in your return to the design in respect to God the designer. Mm-hmm. What essentially you must go to, I think, with that is that death is at the heart of the coming of life over time. And it seems to me you've got a problem of historical Adam or where does sin come, but actually more deeply, the problem that Christian theology has to deal with is, which is Paul's deeper problem, is what's death and where does it come from? Mm-hmm. And to tease our minds around the fact that death is the fabric of God's designing element is going to throw us for any thoughts around this? I mean, what do we do? And what is death biologically speaking then in this case?
Great question. Thank you. It's a very common question. You know, death is presented in Scripture as the enemy that is defeated at the end. In Revelation, we see John's vision of the new heaven and the new earth, and there isn't death. You know, tears are wiped away, wrongs are put to right. What's interesting, and actually this steals a little bit of thunder from John Walton, but what's interesting to a number of theologians is that in the garden, we have a tree of life. Why do you need a tree of life if you're dealing with immortal beings that aren't capable of dying? And this has actually been something that it's not unique to John. Other theologians and Jewish scholars have noticed this as well. So one sort of hypothesis for why that's the case is that humans and other life, as part of the original good creation, that death was actually part of that original good creation. I know it's a little bit hard for us to kind of, I'm not used to thinking about that. When Adam and Eve are expelled from the garden, God says we need to block their access to the tree of life, because if they reach out and take it now, they will, they'll live forever in their compromised state, which would be hell on earth forever. So the idea is that perhaps the reason why there's a tree of life is that when we have fellowship and access to God and communion with him, we have access to the tree of life. And when we were expelled, when humans sinned and broke that covenant with God, then that removed us from the tree of life. Now, whether one thinks of that as a literal tree or a metaphor of some kind, but we're not in close communion with God anymore. We no longer have access to his life-giving presence. And then the natural result of that is the death. But now it's not a good death like it would have been in the original creation. Anyway, stealing a bit of thunder from John, he can get into it more if you're interested in those books, but fantastic question, and thank you for it. Yes, sir.
A comment. I've been waiting for this lecture for at least a decade or two. Thank you very much.
You're welcome.
One question, there are oodles of them, but if the Homo sapiens have developed through an evolutionary process, then they never attained a state, a perfect state. They never attained the height from which to fall. So what do we do with...
What does one do with that? Yeah. This, again, kind of goes back to theological questions. What does God say about the creation? It's good. And then after humans are made, it's very good. Now, I'm not a Hebrew scholar, but I try to read folks who are. There is a word for complete or perfect in Hebrew, but that's not the word that God chooses to use. He uses good, very good. So sometimes other scholars and I too sort of share this concern that sometimes we take our idea of what the pre-fall state might have been like, and we think perfect in our sort of understanding of perfect. But that actually might be pushing the Hebrew farther than the Hebrew will actually support. So similar to the previous conversation where we had, could death have been a natural death after a good long age? Could that have been part of God's very good creation at the beginning? Could animal death, plant death, could that have been part of the original good and very good creation? So that's those kinds of ideas that we end up thinking about. Yeah. Good, great question though. Appreciate it. Other questions?
I can go along with things in terms of the general picture, but a place I always get hung up is what happens with birth defects, especially chromosomal defects that lead to a lot of misery and a lot of pain.
Yeah.
Are they just mistakes in the process? A slip of the knife. I do woodworking, and so sometimes the chisel slips and I cut something I didn't cut off, a piece of wood that I didn't want to.
Yeah.
Do you have any comments on that?
Yeah, not easy questions. The broader question is sort of the problem of evil, is what it kind of really comes down to. It's a subset of that. Sorry?
Is that evil?
Right, yeah. Natural evil, that kind of thing. Why is it this way? And as a biologist, I kind of kick that around, and I think, evolution has as its basis the probability for chromosomal change over time, for mutations, for changes over time. As near as I can tell, the alternative to that is a completely static universe where things don't change, ever. So, because as soon as you have differences, heritable differences, then there's a possibility for differential reproductive success of those variants, and then evolution is going to take place. You wouldn't be able to stop it. It would happen. So it's a question of the alternative seems to be a situation where there's never change. We're asking sort of deep questions of theodicy at that point. Why did God choose to do it this way? And I honestly don't claim to understand that. It seems to me, at least from my limited perspective, that God preferred a creation that had the possibility of change. But part of that possibility of change, at least in our fallen situation, and how does the fall connect with all of that, I don't know, that also leads to the possibility of genetic variation that is harmful. So yeah, it's a challenge. It really is. The broader question is, does evolutionary biology sort of exacerbate the problem of evil is another kind of conversation you could have. The problem of evil is actually something that is a part of any sort of Christian explanation for why things are the way they are. Now, I will grant you, young earth creationists have a very clean theodicy. Everything was perfect. Adam messed up. Well, actually, Eve messed up, but Adam gets the rap, right? Why? Isn't that interesting? It's never Eve, it's always Adam. But Eve sinned first, but anyway. Not that, but that's the narrative of how it's presented, right? So why is Adam always singled out? Well, Adam is the forefather of us all. It has to do with their ideas about biology back at that time and reproduction. But anyway, absolutely perfect. Adam sins. Because of that one sin, all of us live with those consequences. Well, there's some theodicy questions around that too, right? I agree, it's a nice clean theodicy. It doesn't seem to match up well with what we see in natural history. So one then has to weigh how much weight is one going to put on what science has revealed to us versus a particular theology that's developed around Genesis. So, but yeah, you're right. There's not, at least to my mind, quick and easy answers there. Great question, though.
Thank you, Dennis, for a very interesting talk. I'd like to hear your comment on a subject that we're starting to hear more about especially about Mennonites. Most of us have a tradition that went from Northern Europe to Russia to here. Many of our families experienced severe trauma. Of our grandfathers or our grandparents and people that experienced that didn't want to talk about it. There's a line of thought starting up, I think there's even a name, epigenetics or something, where this trauma response can be genetically translated, inherited from one generation to the next. Mm-hmm. And hear your comments on that.
Yeah, sure. And you're right. The name of the field is epigenetics. And what epigenetics is that DNA sequences in certain environmental conditions. The sequence itself doesn't change, but there's actually chemical sort of modification or addition to the sides of the DNA chain based on different environments. And that can actually control in the offspring how certain genes are used, sort of like the copy from the father versus the copy from the mother, which ones sort of take precedence in the offspring. So it seems to be a sort of short-term 1, 2, maybe 3 sort of generation patterning that is added on top of the chromosomal sequence. The best example that I know about, and maybe it's just my Dutch heritage, so I was kind of interested in it. During the Nazi occupation of Holland, of the Netherlands, during World War II, the winter of 1944 to the spring of 1945 was a very, very difficult time for the people under the occupation. There was very little food available. There's a name for it in Dutch called the hunger winter. Children who were in utero at that time, we can pick them out of a population now by identifying repeatable genetic sort of tags that have been added to their DNA in specific places. And I believe, I'm stretching myself here, I believe there's evidence that also may have been transmitted one more generation, although I'm not 100% certain about that. Epigenetics isn't my field. So yes, we've noted it as a phenomenon. How exactly it plays into the biology of the next generation is just something that we're starting to try to understand. We've kind of observed, yes, this is a phenomenon. We don't exactly know how it might impact the next generation. And it seems to be a fairly short-term mechanism. So as such, we're not yet at the position where we're thinking that it's a mechanism that plays much into evolutionary processes over a longer period of time. But it is a fascinating mechanism, and we're just sort of at the cusp of trying to investigate what's going on there. Yeah. Great question, though. Thank you.
Alrighty. Hello.
Hi.
So I suppose that the question I would ask is a little bit similar ties into a couple of other questions that were asked. So it's kind of with regards to salvation and heaven, because as from what I've understood, like, even in a class here, we learned that salvation, God restored His creation, and so if creation was never perfect, then what is this restoration like?
Yeah, great question. We get different pictures of what the new heavens and the new earth look like in Scripture. We see things in the prophets like the lion lying down with the lamb, things like that. We see like the imagery like we talked about in Revelation from John, where he talks about, there not being any tears. We also see imagery like there won't be any more sea in the new heavens and the new earth. And, well, where does that come from? Well, a little digging into Hebrew context gives us the idea that the sea and the ocean is an image of chaos and uncontrolled processes. Part of what's going on in Genesis, like, in Genesis at the very beginning, it talks about how the earth was formless and void and God is presiding over the waters. That's chaos. And then God brings order out of chaos. So part of what's going on in Genesis 1 is that ordering of chaos. Some of what's going on in Genesis 1 is that Adam and Eve are tasked with their stewardly responsibility. And part of that, again, I'm stealing a bit of thunder from John Walton here, is that part of their job is to increase order and to sort of push the chaos back. Not that the chaos is completely absent, even at the beginning, this is John's argument here, but that they're tasked with partnering with God in increasing His domain and pushing back that chaos so that there's order and functionality and things are in their proper relationship. And I think what we see in Revelation is that God is saying, and now we have the final culmination of that activity that's been taking place all along the scriptural narrative. Not that there was complete absence of chaos in the beginning that we're now returning to, but that God's project of having a people for His name and having space that is, John would say, sacred space, that order and God's rule is prevalent in, that has finally reached its final culmination. And that the new heavens and the new earth, there's no more sea, there's no more chaos. It's completely done. All is ordered the way that God wants it to be ordered. Fantastic question, though. Thank you. I think we've got time for one or two more at least. Yeah, one coming here. Okay, great. Yeah, wait for the mic. Yeah,
Sorry.
it's all good.
My question is more about genetics, maybe. So it has to do with human evolution. Now, do we have evidence of possible human evolution since humans started? And what would be the theological implication for that?
Yeah, great question. Is evolution still continuing?
Yeah.
And if so, where's it going? And what might it be producing? Yeah, it's a little bit like asking, is English still changing? Right, if we go back to that language analogy. And yeah, it is. It happens slowly. And evolution, biological evolution, is a lot slower than language change over time. But yeah, as long as there is variation and there's differential reproductive success of that variation, then average characteristics of a population will shift over time. There was a study that was done a little while ago that kind of got some press, and a few people kind of poked me with it because I'm Dutch. And they said, there's the study of, Dutch people getting taller over the last few hundred years and the variation that underlies that. And then my wife says, yeah, why'd you miss out on that, right? So yes, average characteristics do change over time, but it's a very, very slow process. So trying to predict in advance what sort of route it might take is very, very difficult. So
So do we see, like between the first human about, you say, what, 50,000 years ago?
Yeah.
And today, do we see some possible
some differences.
changes that
Yep.
would go along that line
Yep.
of evolution?
I mean, the Dutch getting taller is kind of one
Yeah,
example.
that
That's a
would be
very
one.
short scale thing. We see anatomically modern human remains in the fossil record at least back to 200,000 years. And possibly, I mean, of course, it's going to get blurry, right? The more detail you get, the more you're going to get those lines on a gradient. But back to 300, 350,000 years, we're seeing things that, yeah, they look like they're in our lineage. Are they fully human? You know, are they fully anatomic? Well, it kind of becomes a little bit of a judgment call. Yeah, but we've seen some small changes over that time scale. As we look farther back in the fossil record, of course, we see larger changes. Looking forward to the future, predicting what those changes might be, it's really difficult.
So I guess, I'm sorry, I'm asking a few questions there, but would you say that Adam and Eve were somehow more primitive than humans today?
No, I wouldn't say so, yeah, no. If you took somebody, a Homo sapiens from 100,000 years ago, or even 200,000 years ago, and you raised them from infancy in a current culture, I don't think that they would be distinguishable from any one of us in our population, yeah.
Okay, thank
Yeah,
you.
great question though.
I thoroughly enjoyed that. So I have a more simple question and then something a little bit more maybe out there. Maybe I'll start with the out there one.
Sure.
The Newtonian physics and how this fits in with there a couple of times.
Yep.
But we live in a quantum verse,
Yep.
and I was wondering what the implications were for that, because in that scenario, all bets on Newtonian physics are off. And the more simple question is that with your vertelagen genes, if I'm pronouncing that right, with the egg yolk.
Oh, vitellogenins, yep,
Yeah,
okay,
thank
yep.
you. The homology, the genetic homology looked like it might have been 5 to 10%.
Oh, it's very
At best.
fragmentary, yep.
Right.
Yep.
I was wondering how that compared to the whole genetic homology between a chicken and a human.
Oh, good question. Can I do the second question first?
Yeah, well, that might be preferable.
Sure, yeah, okay. Like absolute homology or absolute sequence identity between chickens and humans, I don't have that figure ready to hand. It's much, much higher within protein coding genes, just sort of like you saw on that diagram. So at a genome-wide level, it would probably be quite low. For individual genes that have had conservation over evolutionary time, it would be much, much higher. So it would be dependent on what part of the genome you would be looking at. What the actual figure is, I don't know. It's been a long time, 310 million years, long time. On the Newtonian versus Einstein quantum physics world, yeah, absolutely. There was a time when Christians actually built a fair bit of theology on Newtonian physics in certain quarters. You know, that the universe is deterministic. If you could wind the clock back and you knew all the forces in play and you had perfect knowledge of all of the molecules and atoms and how they interacted with them, that you could, at least in principle, predict everything that took place, sort of a clockwork universe. And then Einstein came and messed that all up. Absolutely, yeah. Sometimes students use that as an example of, oh, well, here's where we had to completely throw out a paradigm, right? Newtonian physics is completely bunk. But it's not. We still teach Newtonian physics. It works very, very well for things that aren't really, really small and moving really, really fast. But Einstein is a better understanding that includes Newtonian physics within it. Yeah. But you're right, it raises all those sorts of questions about divine action and determinacy, and is it, you know, are there unknowns to God as part of that? My colleague Arnold Sikkema has done a number of talks, and actually on the CSCA website, on their YouTube channel, there's a video of him. He went and spoke at a number of different CSCA chapters on quantum physics and God. So very useful. And I'm not a physicist, so I'll happily
definitely look that up.
punt to him.
Thanks.
Yeah, absolutely. Another good reason to go to the CSCA website.
These are two comments, and I wanted to get your take on them. Trying to come to terms with Genesis chapter 1 in the light of our current state of knowledge. One comment I heard was, we shouldn't focus on Genesis 1. We should rather go to the Gospel of John, how John introduces creation. In the beginning, God created. For the unscientific mind, and for myself with very limited knowledge, that is an easy thing to accept. God created. Now, another comment I heard, which I found very interesting, was that there's a tendency, a valid tendency, to take Scripture literally. But in fact, if we look at Scripture, there's many different forms that Scripture takes, like parables, analogies, things of that nature. And one explanation given for Genesis 1 is that don't take this literally. Basically, it was written to say God created all these things in poetic form without taking it word for word literally. I'm just wondering what your comments would be to that.
Good. I know I sound like a walking advertisement for John Walton at this point, but I have found John's work very useful because the question in my mind is, the best exegesis and interpretation always tries to take into account the original audience and to do our best to understand what they would have taken away from the text. John would go so far to say is that, if it wasn't something that was communicated to the original audience, then, but it wasn't in the first instance written to us in our culture. So he says we have to do the hard work of trying to sit in the audience and take our place in the audience. And I think when we do that, we realize that our Western scientific way of trying to read it as a literal historical narrative might be well afield of what the intent was. Yeah, absolutely. What's interesting is the patristic fathers have their take on it too, right? Augustine's literal interpretation of the literal meaning of Genesis, very different than ours. Yeah. Your comment on John, on the John 1 is also spot on too. There's a number of different creation narratives in Scripture, right? There's one in Job, there's one at the beginning of John, there's Genesis, right? These are not intended as contradictory. They're intended as windows into the picture of what's going on. I think we'll have this as perhaps the last question. Great, and it's a student, so fantastic.
This is less of a technical question, but more about your experience of integrating science into the church. Given that you were talking about how God is both the designer of the parameters of evolution that leads to creation, but also can act supernaturally. Have you found that the supernatural aspect of God has been, or the view of God in churches has been harmed by the integration of science? And if so, how do we need to be cautious in integrating science? I just wanted to know your experience with that.
Yeah, really good question. I think like when you say that, has this caused any damage, a further scientific understanding, and does it, take away from understanding the supernatural aspects of God? I think a big part of that is our natural tendency, because we're products of a Western scientific culture, we kind of have this baseline, maybe unspoken assumption working in our heads that when something's explained scientifically, that we think somehow that takes God out of the picture. And I think we have to realize how profoundly unbiblical that view is, right? It's not a zero-sum game. If that was the case, then science would be among the most blasphemous things to take part in, because every time you explain something through the process of science, you would be taking something away from God. That is also very foreign to the original, at least in Western Europe, how science develops. Science develops because individuals are convicted that nature will have order and regularity behind it because they believe that God is the creator. So they're expecting to find regularity, and that creation will not just be this sort of randomized process because they think that there's a mind behind it. So part of the answer might be reminding us as Christians that we don't view so-called natural processes as somehow antithetical to God's special divine action. Part of the answer, too, is that I think we've done a lot of educating of young people in the church that Well, why do you believe in Jesus? You believe in Jesus because of the Bible. Why do you believe in the Bible? Well, here's all the reasons that the Bible is trustworthy. And one of the reasons why the Bible is trustworthy is because it provides a better explanation for creation than that nasty evolution business. So if you have that kind of as a package, and then you start talking about, oh, there's good scientific evidence for evolution, what you're actually starting to do in many people's minds is starting to try to like unravel their whole view of Scripture and the reliability of the Bible and Christianity. I sometimes remind my students that if we're going to be Christians, first and foremost, the place that we need to start is where Christians start, and that's with the resurrection of Jesus. Why are we Christians? Because we believe that Jesus Christ is raised from the dead. Why is Paul a Christian? Not because he had a certain view of Genesis. He certainly had one. He was a good observant Jew up to that point, killing Christians. Then he has an encounter with the risen Jesus, and he realizes he has to change everything. Everything has to reorient around that realization that Jesus is the resurrected Messiah. Now, what does that mean? And everything flows from there. So it's pretty natural for us to start kind of our Christian education, like you start at the beginning is Genesis, let's start there. I would recommend that we start where Christians should start, with the resurrection of Jesus, and make that the primary focus. And then recognize over the history of the church that there have been many different interpretations of Genesis, like we saw with Augustine. And realize, and another resource that I'll mention again is that sermon that my pastor Dave preached. He talks about how there's different levels of belief, and some of them are up to personal choice, and others are closer to the core, the resurrection of Jesus and the. So I think sort of a reorientation of our thinking in that direction would be of great help in this conversation. Fantastic question, though. Thank you very much.
So just by way of closing, one more time, I'll say thanks to all of you for coming out. Thanks to Dennis for spending the last 3 days with us. Again, it's been wonderful to have you here, and we wish you many blessings on your trip back.
Thank you very much.
Thank you. If you'd like to stay and mill around for a little while yet, you're very welcome to do that. Have a good night.